From Microbe TV, this is Office Hours for Wednesday, May 21st, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you get science, no lies, no misinformation, pure science. And so tell your friends, it's a fun place to be. No question is a dumb question. We just do science. Welcome, everyone. Glad to see you here. Let me thank our moderators before we get started. And the rest of you, tell me where you're from. I want to shout you out. Love to see how many places all over the world people are from. So mods tonight, Vanity Nutrition. Thank you so much for being here. Les is from, Vanity is from Long Island. Les is from California. Thank you, Les. Who else is here? Tom Steinberg from Western Oregon. Thank you, Tom. Always good to see you. I'm heading to ASV in July, and I always think of our ASV where we talked so much. It was just great. Loved it. And thanks, everyone, for coming tonight. Looks like a thin crowd tonight, but that's no problem. What do I have in store for you? I'm going to do some news. Right? This is becoming a de rigueur thing to do some news. And then we'll get your questions and we will do a mini lecture. Today's mini lecture, Antivirals Part 1. I think a lot of you have not taken my course online, so it's okay. And it's a different vibe here to talk about this, right? So it's okay. Now, let me see. What did I want to tell you? Oh, so... Where's my phone? Last week, someone asked me what I'm reading, right? And I couldn't think of anything. In fact, non-science reading, right? In fact, I haven't read very much in a long time. I would say since last fall. So what I did was to go to my Kindle, which is on my phone, of course, and that's got a list of stuff I've read. And I'm in the process of reading Zen and the Art of Motorcycle Maintenance. Now, Urge Condit recommended this to me on Twiv last year. And so I picked it up for some trip and started reading it. And then I just stopped reading. Okay. So that's one of the things that I am currently reading. And I kind of like it because, you know, the main character was a college professor and he has a lot of wisdom about teaching, not so much research, but teaching. And I do love teaching, of course. So anything I can learn about that is great, right? And I also see I have the Daily Stoic. So these are like daily meditations on wisdom, perseverance, and the art of living. So basically how to not let things bother you, right? Which I need to work hard at. Things bother me all the time. Then a lot of police procedurals, which I've read by Mai Showell and Per Walu, two Swedish authors. Tremendous. They wrote 10 police procedurals, and they're just fabulous. They take part in Sweden. A lot of Haruki Murakami, who I love. Oh, my God. You know, I think part of it is the translation. The translation from Japanese to English, I don't know if the Japanese is the same, but in English is so calm. The narration is just so calm and pure. I love it. So lots of Haruki. Then someone told me I should buy Alice Munro and Tessa Hadley. So I bought a couple of books of theirs, and I get a little bit into them and I get hung up. I don't know why. I think it's too sad in many ways, you know, because it's real life. Real life can be pretty sad. People have hardships and stuff like that. No, I know it's reality, but so that's what I'm reading, that kind of stuff. Right now, I've read a lot of other things, but so I do read. And I started Zen for some reason the other day on the train. I opened it up and I started it, and I really got into it again. They took this motorcycle trip to Bozeman, Montana, where he used to teach, and Now they're hanging around there, he and his son, and I really like that. So, all right, so let's see who's here today besides our intrepid mods. Les loves Offit's podcast. Thank you. It's short, right? Sweet, and it's got some clips in it which are really cool. Wayne is from Urbana, Illinois, 57F14C, and pluvious. So today it pluvioted again. It's not a word. It rained again, and it is amazingly cool. It's 11C and cloudy with light rain at the moment. Really, the temperature has dropped. MN is near DC where it's raining. SF, Kip and Laura from San Francisco. Hello, Vincent. RFK and his boss speak as men who have warped reality into their own needs. People like us will not be swayed. I think the people who come here every week are science supporters, are science lovers who realize that science is important for our well-being, right? And so the key is for you to spread the word to others, right? So thank you for your support, Kip and Laura of Science Communication. As you know, Microbe TV runs on donations, so we appreciate it. If you like our work, donate. You can donate here, or you can go to microbe.tv/contribute. Leo, welcome from California. Nice to see you. David Riggs is in sunny and 68-degree Seattle. Kang is from Chicago, 11C and very cloudy. Hello, Questella, Ohio, Iowa-ish. Rima, hello, friend. Hello, friend back. Hope you are well from Iowa. Noir is in Philadelphia, 51F and damp. Good to hear from you, Noir. Oh, thank you so much. We're so lucky. I'm happy. I am lucky that you spend time with me, because without you, this wouldn't exist, right? What am I going to do? Talk at a wall? Because you guys stimulate me. You ask questions, you challenge me, and that's what makes a good class. It's what makes a good live stream, in fact. Helen is in Pittsburgh, where it just stopped being pluvious. So we used pluvious on Twiv a couple of weeks ago. It was in one of the papers. And now I love how people pick up on it, because we picked up on it. We remarked, well, what is this word? And I love how people pick up on it. It's so cool. Brian is in Montreal, where it's 13C and cloudy. Hello, Brian. We are dangerously close to the porcelain in the city. Oh, brotherly love. I'm sorry. I know what you mean by that. Yeah, Leo. We do have another mini lecture today. Carol, our favorite nephrologist from Bakersfield, California, where it is 33C. The heat is on. Yeah, that's warm. Spread some over here. Dean is in Wisconsin, 50F and light rain. Mi Verdi is in France. Nice to meet you. Mi Verdi. Bridget, hi there from Southwest Indiana, 22C and North tornadoes in the forecast. Good. I'm sorry about those tornadoes. We don't get them here very much anyway. Jan Lasko is from Tampere, Finland, 7C and damp. I was just looking at a map of Finland today. Now, why, you might say? Because I was trying to figure out how it was in relation to St. Petersburg, which is really close, right? And then I saw Helsinki, and then not too far from Helsinki is Tampere, and then north is Jyväskylä, where I went last summer to a meeting and did a podcast. A lot of fun traveling to do science and connecting with scientists. Rima and Luana are from Iowa. That's right. Hello again, Elizabeth from West Virginia. Elizabeth came to Twivo live stream this afternoon, which was a lot of fun. We revisited Gregor Mendel and his peas. How cool is that? EHS Talent is in Lehigh Valley, PA. Logotic is in rainy Chicago, 10 degrees C. Mike and Lori in Seattle. Hello. Environmental had a really bad virus last week. Not COVID, nor A or B, no idea. Well, it could have been metapneumovirus, which is what one of our listeners over on Clinical Update had. He thought it was an extended COVID, but it turned out he had COVID, it was cleared, and then he got a metapneumovirus. So it could be something else, or maybe respiratory syncytial virus, right? Nova Rose from PA, a new name, I think. I haven't recognized that before. Welcome to Office Hours. Eliza B is in Toronto. 10C, lots of pluviosity. I've really been liking Dr. Office Beyond the Noise segments. I have to thank Chris Condayan, or Chris Suspect, for his great video editing. He's the one that sticks in the clips. By the way, he used to be in a punk rock band many years ago called Suspect Device. I just tell you that because a visitor to the incubator, I told him that, and he said, "Oh, I'm into punk rock. I'm going to check that out." Sandy, yes, environmental Sandy. I got to remember Sandy. GSD Bio Jonathan, hello 23C, and beautiful riding home from the lab again. That's about what you do at this hour, right? Markle, east end of Long Island. 9C. It's pretty cold. Yeah, you got a little chillier because of the water there. Yep. I am Sandy. Hello, Artemis from Montreal. 6C this morning, 16C this evening. Zen in the Art saved my life way back in the day. I'd be interested if you want to share that, Legat. If not, it's fine. But I like it very much. A lot of philosophy there, obviously. John is from Port Townsend, Washington. Hi to all. Hello, John. Nice to see you again. Tom writes, ASV 2022 was a peak experience for me. Vincent was very kind. People were welcoming, some remarkable conversations. Yes, it was a lot of fun. And Tom was surprised at how much time I spent with him. He said, "Don't you want to go talk to your..." I said, "No, I know all of them. I want to talk to someone who I don't really know, who's my moderator, and who I don't see. So let's talk." Leo, here on the Northern California coast, it's crisp and clear last afternoon. With the temp here a balmy 74. See, the sun is starting to set, and the temps will fall, and the fog will come in. So it's like the same thing, right? That's cool. I like that. Well, Les, if ASV is in California one day, you can come too. We get you registered in some way, and you can come to the meeting. Dion Yahaga, former student tuning in from the Bronx, New York. Former student. Now, Dion, did you take my virology course at Columbia? Let me know in what capacity. Or maybe you're just a former student, not necessarily my student. Okay, that could be. That could be. Hello, Steph SF. You can see by the wrench that Steph is a moderator for us. Thank you for being here. And Questella confirms that she is in Ohio tonight. Gary is in Denver, 73F. John, it's 9C in Minneapolis. I love Zen, but it's been a long time since I read it. And a lot of people love it. Rich Condon said, you got to read it. It's great. Really great. And John says, Sophie's World is a novel I think I liked in a similar vein around the late 90s. I don't know that one. Elmug is from Pueblo, Colorado. Welcome, Elmug. Hello, Lise from Columbus, where it's rainy. Yeah, it is good for the garden. Dr. Prasad did a nice video with the FDA commissioner using plain English, explaining evidence-based medicine. We need more of that from all good science communications. I don't know if this is the Prasad that I don't really like. That's the only problem. Jeff, happy Wednesday from Elgin, Illinois, 10C. Cloudy and wet. And Sandy says it's lousy upstate, 49F. Cold, rainy, and chilly, and had a frost, but the veggies are okay in the greenhouse. Fire EMS is in Atlanta, 75F. Just received my TWIV mug. Thanks. Yeah, if you donate, to Microbe TV. Now, I have an office assistant at the incubator, Valerie, who's been there for about a year. And so she does these things. She can do all this stuff that I just don't have time to do. So you make a donation, you get a TWIV mug. Or another mug if you want. Hello, Jill from the lightly pluvious Wilmington, Delaware, 54F. Rima says, bonsoir, le professeur Racaniello. Yes, bonsoir, Rima. Bienvenue. Office hours. Les heures de l'office, something like that. And we'll get into a language thing again. Arnie, 56F from Ottawa. You're welcome. It's my pleasure to do all of this. It is a pleasure. It is not work. It's a pleasure. Hello, Pat from Nova Scotia, 5 degree C. Joseph is in southern Ontario. No respiratory symptoms at all. What are your symptoms? You got a fever? What else? What do they check? They check. Did they do a PCR of any kind? Elizabeth loves Mendel's experiments. I think that was the first real science I ever learned. Yeah, and on TWIVO, we learn how you can apply modern science techniques to Mendel's experiments. It was very cool. Margot is in Phoenix, where it's 101F in Phoenix Metro. Oh my gosh, OMGGGG. It's hot. Remus says, experiment of Pavlov is the first thing I learned about in school. Then I fell in love with Mendel and Mendeleev, from then Koch, then Pasteur. All great historical figures. That's right. In fact, in the paper today, they said that Mendel's experiments may be the best experiments ever done. That's quite an honorific, right? Peter is in Boulder, Colorado, sunny and 77, F25C. Nevitz says, for what it's worth and totally unrelated to TWIV, there will be no June 1 skiing at Killington, Vermont this year. They had to close up early, May 10th. I've skied at Killington once or twice. A lot of blue ice, man. That northeast skiing. A lot of blue ice, unless it snows. And then you got nice powder. Weasel says, rainy here, trying to figure out this new dehumidifier. Jason is from Southwest France. I don't know. The E is missing. Franck. That's great. Welcome. Ale is from Mexico. Welcome, Ale. Good to have you. Yes, Southwest France. One of my favorite places in the world. Used to go there all the time. Will from Changsha. Welcome. 27C and cloudy. Noir wants to know if I have a twin mug. Oh, if I don't, it's easy for me to stick one in. I have the graphic, right? So I will send you a twin mug. Noir, can you just send me an email where you want it sent? Because you're kind of peripatetic. Is that the word for someone who's traveling a lot? And I'll send it to you for sure. I will have Valerie send it to you. Dion took my virology course at Columbia last May. I tried reaching out a while back via email, but wasn't sure if I wrote to the right one. You might have, but you could try Vincent at microbe.tv. So Dion, now you can call me Vincent. Course is over. You're out of Columbia. Good to hear from you again. I just finished teaching the 2025 version of the course. Always a lot of fun. Hello, Weaselbox. Good to have you here. Arizona, windy Asheville, North Carolina. Hello, Michael from Ciudad de Mexico, 24C. Ian says it's both ironic and grim seeing how much things have changed since last year and our conversations concerning RFK and his campaign run. Indeed, we were worried about it last year. Heather is in Kamloops, 18C, Canada. I drove through Kamloops once. I didn't stop. Maria Fernanda Monsive Lopez. Greetings from Mexico, Monterrey, Mexico. I have a couple of questions about a virology project. Would it be okay for me to send my questions to your Columbia email? Sure. If you have it, you can do that. I'm happy to help out. Leo says, my son and family live about 100 miles north of Mendocino in the city of Eureka. His whole family has been sick for the past 6 weeks. They have been seeing their doctors, getting, next one, no specific diagnosis. Last week, they took the older child back to the clinic. They had an out-of-town physician. PCR results came in as pertussis, no specific diagnosis. Where's the next one? Whole family has been vaccinated. There's a pertussis hotspot in Eureka. Is it fair to assume the vaccine is efficacious and prevents hospitalization and death? It does. But it's quite a number of shots, right, starting at a very young age. So I don't know how many they do for a situation like this to quell an outbreak. But yeah, the vaccine is good. Daniel is always talking about how, a lot of these individuals are not vaccinated. Tom says in TWIV 1217, thinking about a recombination in plus RNA viruses, the RDRP complex and templates must do an intricate dance. I've seen some high-level papers, knowledge gap, RE, all those you-know-whos. Well, the idea is that the RNA is on membranes, and so the molecules are very close together, and so polymerases can skip from one to another as they're copying and make recombinants. By the way, for extra credit in my virology course, I give them, and Dion knows this because he was in that course. I say, watch Contagion, the movie, and summarize what's going on, and then tell me 3 things that are scientifically wrong with Contagion. And actually, most people don't get them. They get other things that are okay, but it's a good effort. But one person had the following answer for one of the things wrong, and he or she is absolutely right. So in the movie Contagion, the virus, what is it called? MEV1, meningoencephalitis virus 1, is supposed to be a recombinant virus between a bat virus and a pig virus. Wrong. It can't be. Negative strand viruses do not undergo recombination. Okay? It's known. They do not. Positive strand RNA viruses do, but not negative strand. And MEV1 was a negative strand virus. So there's another potential answer for what was scientifically wrong. And shame on you, Professor Lipkin, for invoking that. Okay. Noir, thank you for your contribution to science communication. Really appreciate that. Send me your address. We'll send you the twin mug. Profelo Mateo, how's everything? Yeah, everything is a big, I know it's a colloquial expression, right? So the Microbe TV is great. We're cranking out podcasts. We're trying to reach more people and give them good science. I taught my course. It's great. But the state of the U.S. is horrible. And so I can't handle that. It's terrible. And this is not a pod where we're going to talk about most things other than science, but everything is getting screwed in this country. And I can't wait to get this MAGA Republicans out of there. What do you recommend for someone who wants to begin with sycom? I think so. The way I began was blogging. Blogging is free. Don't pay. Don't pay to blog. You can find lots of free platforms. Um, and you get practice at writing and addressing a bigger audience. And you can build up that audience. So that's what I did, and I built up an audience. And I had the advantage of having a pandemic and a couple of outbreaks there, so people came and wanted to know. But I think that's the best way to start. And then, of course, I segued into podcasts, which is a little more involved. So I wouldn't start with them, although they are appealing, right? Because you like to talk, you like to record, especially if you want to do video. People can see you. You can have a YouTube channel. If you want to do that, that's fine. But I would recommend blogging first. Hello, John from Thailand, where temperature is irrelevant. It's so high, right? John says, speaking of skiing, I learned from TWIM that some snow machines use bacteria to make use of processes gleaned from microbiology. Yes, nucleation bacteria to make ice. Very cool. You're talking about MEV1, the contagion virus? No, it's just... Yeah, they didn't say it was multi-partite genome. But you're right. If it was like influenza viruses, reassort. So segmented negative strands reassort, right. But they don't do the recombination. Yeah, good point. Hello, Aubrey L. from Richmond, Virginia, where it's 63F. Noir, this is antibiotic resistance to bacteria's own adaptive immune system. No, it's not. Actually, what happens is the genes that confer antibiotic resistance are typically, say, on plasmids or other mobile or phages or other mobile DNA elements, and they enter the bacteria and they confer antibiotic resistance. Now, remember, antibiotic resistance didn't come about because we used antibiotics. In nature, bacteria encounter, say, in the soil, other bacteria that are making antimicrobials, and so they have to be resistant to them, particularly fungi or others. But so antimicrobial resistance has been around long before we started using antibiotics. But it's not an adaptive immune system. It's just a gene that confers resistance that they get from other sources, other bacteria. Love that you use Contagion as a teaching tool. Yes, because there we have a blockbuster movie about a virus, right? How could I not? And there are lots of movies about viruses, actually, but most of them suck. Contagion is reasonably good, although there are scientific inaccuracies. And, you guys, if you want to take guesses, that would be cool. Hello, Privyet, Veronika. Welcome back because you were at the Twivo live stream. Cool night, 10C in Moscow. David is a recently retired optometrist trying to use my background to help with virology and eye care. Can you explain why measles and COVID have an affinity to conjunctival cells and influenza viruses too? Many viruses like conjunctival cells, the outer layer, right, of cells in the eye, and lining the eyelid. So they're very exposed, and they have receptors that the viruses need to get into cells just coincidentally, and so that's what they need to get in. And then, of course, the cells are hospitable to support viral multiplication. So many viruses. I have a great slide of an eye showing all the different places where viruses reproduce. It's in Infection Basics lecture of my virology course, which you can find on YouTube. Environmental. Sandy, at my former workplace, Buffalo U, they chased a graduating senior off the stage by police for holding a baby dressed in a cap and gown. Yeah, our country is nuts. It was cute. I don't see what's wrong with that. It's his baby. That's cool. John, I remember someone saying in Contagion, the actor stabs her thigh dramatically with the cure, and it isn't how it should have been administered. Well, John, you took my course, so I don't know if I mentioned it there. So Ali Hextall, who, by the way, I met the actor who plays Ali, Dr. Ali Hextall. I met her, and I have a selfie with her on Instagram. If you want to go check it out, I was starstruck. She's just another person, right? So she develops a vaccine, which she tests in one monkey. Ridiculous. And then she takes the syringe and jabs it into her thigh. And then an hour later goes to visit her father, who is dying of infection. And she's protected. Complete BS on multiple levels. First of all, and then the vaccine is licensed as a nasal spray. So first of all, you don't use one monkey. Second of all, you don't stick it in your thigh and then make a nasal spray out of it. And third, you don't go within hours for a challenge. You need 10, 12, 14 days to make antibodies and T cells, right? So the students don't get that, frankly. They don't get it. I don't know why. Okay, so that's what's wrong with it. And I was at this symposium where Ali Hextall, the actor who plays her, was there. And I got up and I said, yeah, I teach this course here and I give the students extra credit. And I asked them to tell me 3 things that are wrong. She said, what are the 3 things? And I told her, and she didn't know anything about it. So apparently the vaccine was originally, she was going to originally test it intranasally. But on set, the actor said, this is not dramatic. I want to stick it in my thigh. And so Ian Lipkin, the script consultant, was not there. So they changed it without really knowing that it was a faux pas. A faux pas. Yep. Oh my gosh. Carol says, I also can't wait to get this MAGA admin out, hoping the next admin will refute this COVID lab leak nonsense. It's ridiculous. It's just nonsense. I'm glad, Carol, you recognize that, because so many Americans do not. It's nonsense. There's 0 evidence supporting it. Oh my gosh. Costello bought the movie after I gave it as homework some years back. That's cool, Costello. John, I was vaccinated for measles in 1960. Do I need to be vaccinated again? Well, you know what, John? We're going to pull up Dr. Griffin's table, which we can peruse and ask exactly that. So give me a minute here to go to Microbe TV, and we're going to go to TWIV. And unfortunately, the website is up tonight, so I can do this. Okay, Daniel Griffin, and then I'm going to put it on the screen here so we can look at it together. Okay, here we go. Measles vaccine recommendations by age group. Okay, so adults, if you have no immunity, you need to get 1 and 2 doses. If you're born prior to 57, you don't need vaccination because it's assumed you were infected. And if you're born in 57 or later, if you've got, let's see, you were vaccinated in 1960, and sounds like you got 1 dose, so you need to get a second dose. From 68 to 89, only 1 dose was recommended. And so you were, in 1960, they're actually using a different vaccine, so you should get a second dose according to this chart. Okay. Cool, right? Very cool. All right, let's get that out of my way. Andromeda strain. Right, that's the scary one from the 70s. Yep, Bettabacterium. Weaselbox, of course there's scientific inaccuracies. It's the movies, but it's pretty darn spot on. Weasel, I know it's the movies, but I want the students to pick up the inaccuracies, right? That's the whole point of it being a teaching exercise. I understand it's a movie and it's got to be dramatic and it's science fiction, but I wanted the students to pick that up. You can't remember what the 3 wrongs are. Maybe you should go watch it. N equals 1. Sounds like RFK Jr. and Trumpian science, indeed. Jennifer Eel, right. So I met her. You could go to my Instagram. I don't have that many pictures, so we could actually, let's see, find it and show it here. Yeah, I haven't been, log in. Come on. Good. Log in. Always a problem. Password incorrect. Screw you. Maybe I can do it on my phone. Yeah. And then I'll hold it up to the camera, right? Passwords. I mean, you need them. I know. But when you really need to show something, they don't show up. They don't work. Jennifer, where are you, Jennifer? Jennifer. It was over a year ago, I believe. Well, I guess it's not on my Instagram. I didn't post it. How can that be? That would be like the thing to post, right? All right. I will show it to you another time. Sorry. Jennifer Eel, right. I came up to her. I said, can I ask you a question? She said, of course you can. And I said, can I have a selfie? She said, of course you can. Jill says, I love when actual scientists debunk the science in movies. Between Vincent talking about Contagion and Neil deGrasse Tyson picking apart various space movies, it's the best. Yeah. Jill, you think I should do that more? Maybe I should make a video picking apart Contagion. That would be cool. We kind of did it on Twiv, but, nobody goes back to the old ones. Patricia's in Massachusetts awaiting a nor'easter. Sandy says, your channel gives me a sense of normalcy in such an abnormal world. I try to also, but the news in the climate world is distressing, and the U.S. is trying to hide the truth. It's so, of course it is. They always have tried to hide the truth about climate change. And, it's going to hit, and it's going to screw things up really badly. Well, thank you, Noir, for your contribution to science communication. I appreciate your support, Noir. I really do. David says, the bubbas who find the dissolving, suppurating soldier in the stand say it must be something he ate. Ariozone, I messaged you on Instagram today, shared a short YouTube of a fourth grader in New Jersey with autism speaking to his public school classmates about RFK Jr.'s lies. Best criticism I've heard. Oh, this is fabulous. Thank you. I'm going to repost that. Great. Love it. Weaselbox is going to put on Contagion Friday after Princess Bride. Implosive Alloy saw an NPR article that the CDC's communication has been pretty curtailed. It really has been. And I'll have some words about this in a bit. Dorothy says, my partner's doctor did not respond to a gentle request for an MMR booster. Are we asking for the correct product? Yeah, it's measles, mumps, rubella. That's what you need to get, according to according to that chart I just put up. Helen was born in 1975. My PCP said my insurance wouldn't cover a MMR booster unless I was in the 63 to 67 window. So you're born in 1975. Let's go back and look at this figure that I just put up. Born in 1957 or later. So what do you have, Helen? Because if your vaccine history is unknown, you should get 2 doses. And depending, so 75, if you're vaccinated between 68 and 89, you would get a second dose. So you should be eligible. That's not right. You have to get the chart from Daniel's website and show it to your PCP. Homework challenge accepted. So you know what we could do, folks? We could have you guys look at the movie in the next week and come up with 3 things that are scientifically wrong. I want science. I don't want other things. A lot of the students put other things than science, right? No, I want science. And then we'll go over them because I'm going to get a lot of answers that I haven't thought of. So it should be cool. Right? Let's do that. Carol found the picture. 4/12/24. Yes, thank you. You did what I could not do. Well, I couldn't log in. I mean, I'm looking at it on my phone, and I didn't see it. Why? Why not? Professor VRR. That's June. It's not on my phone. I don't know why. Anyway, I'm glad you found it. Kang says a great way to teach science is to ask students to point out mistakes in science in any science fiction movie, for sure. I love that. I'm not saying it's a bad movie as a result. Okay, so there are 2 things. If you need to make an adjustment because of drama, okay. But if you just don't know the facts and you put something out there that you think is right, that's wrong. You should get your facts straight. Because if it doesn't impact the drama of the movie, get your facts straight. Why? Because a lot of people are going to see it and they're going to think that's right, and you've just taught them the wrong thing. Margot also found the picture. Thank you. April 18th, 2024. Thank you. Not on my phone. I don't know why. Alternative science, indeed. Bridget says, between cuts at NOAA, NWS, and FEMA assistance is going to be awful come hurricane season. The brain drain is also concerning. Yeah, absolutely. Challenge accepted. Okay, folks, do you think that's a good idea? It's not bad. You get to watch a movie and just have a little notepad. I'm showing my age, a phone or a laptop, whatever you want to take notes on. And as you're going through, just say, that's not right. And you can list as many as you want. It's just at least 3, since that's the extra credit. And here we go. We're going to make it worth your while. Anyone who gets all 3 right, so I have a list of 3. Anyone who gets all 3 right gets an item of choice from the Microbe TV Cafe Press store. Right? You got mugs, you got glasses, you have stickers, you have pins, you have shirts and hoodies and zip-ups. Okay? Your choice. One item of your choice. And, I'm flexible. If you say something like, I just did no negative strand recombination that I haven't thought of, you still win. I'm fair. I recognize that you don't have to have my 3 answers, okay? Looks like Contagion is inexpensive to buy or rent. Challenge accepted. Excellent. So we're going to have a couple of people doing it. It's great. Also ask students to point the mistakes in the science in this administration's pressers. Yeah, for sure. I mean, the latest recommendations about COVID vaccines, right? 65 and over. And under only if you have at least one condition that is known to lead to severe COVID. This is BS. 200 plus kids died of COVID during the pandemic. They're someone's kid. Why can't they be protected if they want to? I'm not talking about mandates. Why can't they be protected? They're wrong about this. Is that the movie about the monkey that escaped? No, Contagion is not about that. I don't know what movie that would be. Jeff says, I think most people believe vaccines work as fast as the EpiPen. Personally, I hate the collapse of time in movies to fit invented. And that's the problem, right? You can't wait 2 weeks. What are you going to do in the meantime, right? Exactly. Xavier says, I'm watching Contagion in my med tech class tomorrow. Just started your YouTube course. So are there any specific lectures for me to watch to pick up the mistakes? You should watch the vaccines lecture and maybe infection basics and pathogenesis. Yeah. Yeah. Hello, Richard, DDS. Richard, hear from Richard a lot. We appreciate your communication. Is everything okay with Amy? I really can't speak for Amy, because I speak to her on a regular basis, and I don't want to. There's some things I can't talk about and so forth. But as you know, in general, the FDA is in difficult times. They have had their budget cut. Many people have been fired, and it's very difficult to do research there. So, those are the global problems. Maybe someday she'll come and talk about it. Mark says the good vaccine update I saw that really helped me was the Tdap. Needs to be updated every 10 years. My physician did not know it. Yeah, every 10 years. That's right. I'm up to date. I just went for a checkup last week. I'm up to date on Tdap. And I got a pneumonia vaccine. And he said, this is the last one you'll need because it's durable, right? Dr. Larry Brilliant loves Contagion because he was an advisor. And yes, he was at this symposium that I attended where I asked the actor for the mistakes, and Brilliant just kind of sat there and passively, not really that interested. Outbreak, is that where a monkey got out? Okay. Yeah, we haven't given any spoilers here. The only thing I said was the recombination before, because that was really, really a good idea. Hello, Rona. Rona, Long Island. Yeah, apparently the Knicks are doing something good. I don't know about these things, but, the incubator is right near the Garden, MSG, and it's on a playoff night, it's a mess. It's a mess. Monkey that escaped is at HHS, indeed. The best way to protect vulnerable groups would also seem to limit the spread in general, so there are fewer chances for them to get infected, which means vaccinating everyone. So that's the idea behind school vaccine mandates, right? That you prevent spread of diseases in schools. But, that's going to change. I'm sure they're going to get rid of all these. But I mean, some people want vaccines, so I don't think they should be limited because some bonehead thinks they don't work. It's not fair. Hans is in Guayaquil, Ecuador, where it's really hot, 29°C. Outbreak based loosely on Ebola. That's the one with Dustin Hoffman. That's horrible. Outbreak is horrible. I mean, if I reviewed that, it would take hours, longer than the movie. I think that's what people are saying. A monkey escaped an outbreak. Yeah. Dorothy says, please remind Amy that her fans are worried and are pulling for her. I will. Every week I say, Amy, people are talking about you on office hours. They're asking about you. I don't tell them anything, but I want you to know they want to know. Okay? So she knows. Yeah, Outbreak had Dustin Hoffman. That's right. Horrible movie. Yeah, Laurie Garrett was also an advisor for Contagion. Yep. Patricia also wants to know how Amy is doing. Jason, what effect do radio signals and specific frequency patterns have on viruses? I don't think radio signals have any effect. It's other wavelengths of light that, like UV light, right, or infrared. Not infrared, no. But not radio waves. I don't believe that has any effect. Thank you, SRR, for your support of science communication. Really appreciate your donation. Angry Penguin asks, could mRNA technology be used to make antiviral antigen into a vaccine? Yeah, in theory, you could. You just make the mRNA encoding the antigen, you wrap it up in a lipid nanoparticle, and you have to test it. Now, it doesn't work for all antigens. You have to test it. But yeah, in theory, you could make any vaccine. But this administration is very negative on mRNA vaccines. I don't know why. They think they're dangerous, and they're not. They're not. They simply are not. They've been in tens and hundreds of millions of people. And there are some side effects, such as myocarditis, but it's less frequent than after infection, much less frequent. So you have to do the benefit risk ratio. You have to think about it. And, I got 3 doses. I think that it's a no-brainer. Leo says the Contagion assignment will be fun. As everyone knows, I'm not a doctor, but I have played one on TV. Cool. So you have to say what's in the script. You can't modify it, right? If you find something wrong, House was wrong. Dixon always used to point out where House was wrong. And that's the script writers, right? They should know better. Thank you, SRR, for your contribution to science communication. Appreciate it. Yeah, get out in the sun and get some vitamin D. Thank you again, SRR. Appreciate your support of science communication. When I say radio, I mean electromagnetic radiation. I understand that the radio frequencies are a certain cut of the whole electromagnetic spectrum, right? And in there are UV and other things. And so, yes, certain frequencies of radiation do affect viruses, right? So UVC was highlighted on TWIV. We did a program with a guy at Columbia who's working on that to, irradiate spaces. Regular UV inactivates viruses. And it may be that other frequencies do as well. They don't come to mind right now. Yep. Yeah, most people don't understand user risk benefit. They see a side effect and they say, that's it, I'm not getting it. I'm going to get the disease instead, which has a 100,000-fold higher rate of side effects, I don't know what to do about that, Weasel Box. Sandy says, Novavax got approved in the U.S. over 65, right? Or if you're under 65, you have to have an illness that is known to increase the severity of COVID. Yeah. And Noir says, Amy is someone I respect and admire. Wish we could see her presence again. You never know what the future holds, Noir, right? Mark says, some idiot on the Tucker Carlson show yelled about gain of function. So now all mRNA vaccines are bad. I don't know what mRNA vaccines have to do with gain of function. And this idiot doesn't even know what gain of function is, making it all political when it's lovely science and you're turning it into crappy politics. Vitamin D, get that vitamin D, yes. Tryon, hello Tryon. Are viruses electrostatically charged? They are charged, yes. Remember, they have phospholipids in their membrane, which are negatively charged, and so does the cell. So how do you overcome that? You have to bind a receptor that is putatively positively charged, right? And that will then pull it into the cell. UV and ozone, right? X-rays are part of the radio frequency spectrum. Yes, Maxwell's spectrum. Yeah, you got that whole thing there with the different wavelengths. I love it. Yeah, so X-rays will inactivate viruses. UV will inactivate viruses. A lot of people haven't sat through a statistics class and don't understand how numbers work. Well, most statistics professors are not very good, or teachers are not very good. Mine was horrible. I never got it until years later when I needed to use it for my research. And then it made sense empirically. But you're right. David says, I have a colleague with PEG allergy to the extent of anaphylaxis. Pfizer and Moderna contain it, but not Novavax. Yes. Well, if you know that, then you get Novavax for sure. Richard says, can Daniel review the how to approach diagnostic testing when one is seeking care for a respiratory infection? The symptoms are similar. At times, one needs to be one's own advocate. Why don't you send an email to daniel@microbe.tv. I'll put it on the list for tomorrow, Richard. That's a good question. But keep it short. Long emails, like a page long, it's really hard to get that in. Christopher says Novavax is available in Sweden, but our public health can't tell us where to get it and refers to the region, which can't tell us either where to get it. That's great. Come to the U.S. If you're over 65. Red Hen says, next week I'm going to start a clinical trial that includes an mRNA flu vaccines. There are 6 different options in the trial. I won't know if I'm getting a regular flu vaccine, mRNA flu, COVID, or a combo. Well, Red Hen 689, thank you for volunteering. We need people like you to do these clinical trials. And if you're coming back here on office hours, let us know how it goes. Elizabeth says, I'd like to push the seatbelt comparison. You risk bruising with belts versus going through the windshield without. Sometimes it gets through to people. Seatbelts are mandated also for the good of everyone. And it's a freedom you give up, not being able to climb in a car and not buckle up, right? You give up that freedom to protect yourself. And it's the same with vaccines. Okay, Mark, UV spectrum is short of the wavelength. The higher the energy, wavelength longer than visible red have very little energy. And the high wavelength, of course, is what inactivates things. John Briggs says, Dr. Brilliant and WHO teams forcibly vaccinated hundreds at night in India. They had to do it, and it worked. What's your opinion of Jay Bhattacharya? Completely unqualified for this position. He's a health economist. Never practiced medicine. He is one of the co-authors of this Great Barrington document, which advocated just let it rip for COVID, okay, without thinking that there might be other side effects that we don't know about of COVID. And then we found out long COVID, and now as a consequence, long COVID is higher in young people. And so he said, young people are healthy. You can let it rip and make a good immunity. Didn't know what he was talking about. And he doesn't know what he's talking about as head of NIH. Totally not. You can see just a few things he said so far. The wrong person. And did Michael Schmidt have a microwave sterilizer? I don't know. Not sure. Peter S. tells us, thank you, Peter, once photons get to the energy level of the color royal blue, they carry enough energy to break carbon bonds. It's a very mild effect until you reach deep UV energy levels. Thank you. I love this electromagnetic radiation spectrum. I just don't remember it because I don't use it. Christopher, will you do a new episode on far UVC? A lot seems to have happened. We could do that. A number of people have asked for that. Yeah, and maybe I go back to that person from Colombia. That would be good. Jill said, in college we called our statistics class sadistics. Very good. I like that. That's really good. Sandy says, RFK Jr. will find something wrong with seatbelts. Shh. Gosh. Peter says 4 out of 5 statistics account for 80% of statistical data points. Right. Patricia says, would Dr. Offit consider a longer podcast with you? He's awesome. I'm sure he would do it, but my original idea was to keep it short, to engage people, and it's done pretty well. We don't break 10,000 views initially. Eventually, some of them get over 10, but I think the brevity. I try to keep it to 15 minutes or less. He has been on Twiv, right? So there are some longer podcasts, and I could always invite him back onto Twiv. I'm laughing because Ms. Martian says 4 out of 3 people have trouble with fractions. Beautiful. Beautiful. I love it. Weaselbach says, I found giving a short background on how those numbers work, then people were more open to risk benefit. Didn't sound as scary. That's good if you know enough to give a short, simple background on the numbers. Yeah. Okay, let's see. We're going to mark this and we're going to do a mini lecture. We have to turn off Weaselbox here. Okay. Antivirus. So mini lecture on antivirus. We're going to break it up into 2 because play in window. Okay, so play. Why are you not playing? Same thing that happened last time. I have to move this out of the way. There we go. Antivirals. I love the quote here. Look, folks. Though the doctors this is from Leo Tolstoy, long time ago. Though the doctors treated him, let his blood, and gave him medications to drink, he nevertheless recovered. You get it? I love that. All right, antivirals. Vaccines, as you know, can prevent viral disease. We talked about that last week, the last 2 weeks. But they don't really work if you're already infected. Do you know what the exception is? One exception, folks. Put it in the chat. But we have a second defense, and that's antivirals, which can stop an infection after it started, although you can't wait forever, right? You cannot wait until the virus titers are low because an antiviral is not going to do any good. And that varies for each virus. Of course, for a chronic virus like HIV, you can start any time because you have years of replication, right? Yes. Wayne is the first to say rabies, and also Javier said rabies as well at 9:03 p.m. Rabies is the only one where you get bitten and you can still be vaccinated and prevent getting rabies. However, we don't have a lot of antiviral drugs. Here's a timeline of number of approved drugs from 1959 to 2018. And you can see it really didn't get going until the 90s. But most of these drugs are against hepatitis C virus, HIV, herpes viruses that infect you forever, right? So again, it's easy to diagnose and then treat you at any time, basically. So we have about 100 antiviral drugs on the market today. Now, why is this? Well, first of all, what are the targets and the viruses? So most of the antivirals we have, 43, is maybe a little different number, but HIV, Hep C is next. The herpes viruses with 11, HBV with 8, and then influenza 4, and then others. So you can see we have most of the antivirals against a few viruses. The targets, polymerase is a big target. It's an enzyme. Protease is a big target. It's an enzyme. Integrase is another target that's an enzyme. There aren't so many of those. NS5A is a Hep C target. and then 10 host targets, which is broken down here on the right. It's 75 antivirals against the virus and 13 against the host. Host targets are hard because you get more side effects if you target a host function, right? So why are there so few antivirals? Well, the main problem is that, viruses are obligate intracellular parasites. They need host functions. And so often they're related. So a polymerase in a virus is related to a polymerase in a cell. So you make a drug, it will inhibit the cell. And you can have off-target effects as well. So adverse effects. We don't want side effects. And, every step in reproduction engages host functions. Some viruses are hard to propagate. They don't have an animal model. They're dangerous. So HBV and HPV are very difficult to propagate in culture. Smallpox, there are 2 antivirals, but they weren't tested against smallpox. Very dangerous. Can't work with it. Ebola virus and Lassa virus, of course, are dangerous. You have to work under BSL-4. Although there are ways around this, as you will see. But the most important reason that there are so few antivirals is that any compound you have has to block virus replication completely. It has to be potent. Now, most of the drugs you take are not 100% effective. They don't have to be. They work by having a fraction of activity inhibited, like aspirin, right? It doesn't inhibit all of the activity of the target. But you can't have partial inhibition of virus because that will select for resistance. You allow the virus to reproduce in a low level of drug. Now, this is illustrated on the right here. We have viral load on the x-axis and drug given over time. The optimal dose brings viral titers down close to 0, and that's what you want. If you give an intermediate dose, you can see the virus reproduces and eventually overcomes the drug, and a low dose is even worse. And here you're going to have the generation of mutants. So you need to have a potent drug, and it makes it expensive because you have to go through a lot of compounds to get that potency. The other issue is that many acute infections are short. Remember, an acute infection, a defined period of virus production and disease. By the time you feel ill, it's too late to have an impact on clinical disease. Not always, but often. So you have to give antivirals early in infection, a day or 2 days, or with Paxlovid up to 5 days after infection, but no later, it won't work. Now, you could, in an outbreak situation, give drugs prophylactically, but we don't like to do that. We don't like to give drugs to healthy people, with the exception of pre-exposure prophylaxis for HIV/AIDS. So, what that is someone is at risk or is about to engage in risky behavior. You take some PrEP ahead of time and reduce the risk of being infected. Now, if we had broad spectrum antivirals, that would get around this, right? Because you could give that to people as soon as they have flu-like illness. But we don't. We don't have broad spectrums. And, the paucity of antiviral drugs is also because we don't have diagnostic agents. So a company is not going to make a drug unless there's a diagnostic for the infection. And nobody's going to make a diagnostic if it's not useful, if there isn't a drug that would be an actionable item, right? So it's a bit of a conundrum unless you have a pandemic, and then you got to do it. A little bit of antiviral history. The first search for antivirals began in the 1950s. Chemists were modifying antibiotics of the sulfonamide category. They made chemicals thiosemicarbazones against pox viruses because, after World War II, smallpox was still a threat. Then in the 60s and 70s, companies began what we call blind screening to find chemicals with antiviral activity. And they were doing this because, the antibiotics were working well for bacteria. And they said, well, why can't we do this for viruses? So what is blind screening? There's no attempt to focus even on a virus or a virus-specific mechanism. You just take random chemicals or natural products and look for the ability to inhibit replication of a bunch of viruses in cell culture systems. So random chemicals, every drug company has a huge collection of chemicals that they've made for other purposes. They just go through that library and test them. Do they work for flu, RSV, polio, hepatitis, HIV, etc.? Natural products. You go get some dirt, as you can see here, and you bring the dirt back in the lab and you culture it and you take the supernatant and you see if there are any microbes in the dirt that are making something that will inhibit some virus. All right, so those are called blind screens. And then you get a hit on a virus. So then you try and purify the active ingredient. You try and modify them to reduce their toxicity, make them more soluble, make them bioavailable, make them go, if you take them orally, make them go to the place where they're needed, and other properties as well. So that's blind screening. And you make thousands and thousands of molecules before you even get to humans. A lot of effort, very little success. And one exception was amantadine, which was approved in the late 1960s for treating influenza A viruses. That's amantadine there, a weird little chemical. One of 4 drugs available for flu, but not used really because there's widespread resistance to this antiviral. We didn't know how it worked. It was just approved on the basis of clinical trials. But in the 90s, we learned the mechanism of action, which we'll talk about in part 2 of antiviral drugs. Now, today, it's very different. Antiviral drug discovery is very different. We use recombinant DNA to produce specific viral proteins. We use incredibly sophisticated chemistry to make all kinds of compounds. And we can do really targeted approaches. So you can clone genes, express them in systems, purify the product. You can look at the structure. And we know reproduction cycles for most viruses, so we can say what we want to target. And even if you can't propagate the virus in cell culture, you could make a viral protein, a polymerase or some kind of an enzyme, and test it, and then figure out how to get the efficacy later. So no more blind screening, basically. We do very targeted approaches, and we take the reproduction cycle of viruses and we target specific steps like attachment, uncoating, mRNA synthesis, polyprotein processing, nucleic acid replication, integration for HIV, assembly, and release. And we have antivirals, as you can see here, against the variety of viruses at each of these steps. And in part 2, next week, we'll talk about how some of those work. But the pathway to getting a drug is convoluted and incredibly expensive. It's kind of outlined here. You start with a medical need, right? You have a virus that's causing substantial morbidity and mortality. And then you do some research to identify a target. What are we going to inhibit? And then you do some screening. You can do all kinds of screens, and I'll give you some examples of those in a minute. You get a lead compound. You modify it. You want to know things like, once you get into animals, you say, where will the compound get to the right place in the body at the right concentration? That's bioavailability. Will the compound persist long enough to be effective? Pharmacokinetics. Will it be safe, right? Toxicity and specificity. And so your lead is checked and modified and checked over and over until you have a drug candidate, which goes into what we call preclinical studies in animals. That's preclinical. And then eventually in humans. And of course, if you have a medical need, it's urgent, right? So there's a pandemic of a new virus. You don't have the luxury of spending a lot of time to do this work. So you have to compress it. And here's a typical timeline for drug discovery. So, you have your discovery and synthesis 5 years, your preclinical another couple of years in animals. Then phase 1, it's a safety assay in 20 to 80 healthy subjects. Phase 2, efficacy. Is it going to reduce disease or progression to hospitalization? 100 to 300 subjects. And then phase 3, efficacy, side effects, superiority over if there are other drugs already licensed. Yours has to be superior. And this can be many thousands of subjects. So typically we're up to 12 years, and then we have an FDA review. The product is licensed, and then it undergoes continuous surveillance for safety, which is sometimes called phase 4. So a long time, you can see here, it takes to get a drug to market. Now, Paxlovid was out within a couple of years, right? So research began immediately. There were some leads already on other coronaviruses, so that helped. But it was very accelerated. Okay, so how do you screen for antiviral drugs? Well, you can do what's called a mechanism-based screen. So let's say you want a drug that inhibits a viral protease. And so you have to know the cleavage site of the protease. So these A, B, C, D, these are amino acids. So you need to do some research to know the cleavage site. And then you do the screen, which involves having a fluorescent or some kind of marker that you can assay attached to one end and then a bead at the other, so you can remove the material. And then if you add protease, it will cleave it, and then you give a soluble material that is, say, fluorescent, and an insoluble material that can be removed from the solution. And then you can add the drug and see the effect on liberation of this soluble material. And so here is an assay where we're looking at fluorescence intensity of the soluble peptide here that would be released after cleavage in the absence or presence of drug. And this is in minutes, 250 minutes. So you can see without drugs, the cleavage just keeps on going until the precursor is exhausted. But with drug, you inhibit the reaction. And so that can give you some candidate compounds that you could modify further and get better activity. And you want to have nanomolar effects of inhibition, not micromolar and certainly not millimolar. You need nanomolar. Now, cell-based screens are cool. Here we use a bacterium, or could be a yeast or other cell, but this is illustrated in bacteria. And what we're using is tetracycline resistance. So that's an antibiotic which will inhibit bacterial growth. And the resistance is conferred by this protein, multi-membrane pass protein. So here's the membrane of the bacteria. And this is basically a pump that pumps the tetracycline out of the cell, and that's the basis of resistance. So if you add TET to the culture, this pump will pump it out so the bacteria are not inhibited. So what you do is you say, let's say we're looking for HIV protease inhibitors. You engineer the HIV protease into one of these loops that are in the inside of the cell. And so if you express or produce the HIV protease in the cell, it's going to cleave this loop. And now these bacteria are going to be sensitive to tetracycline because the protein, the efflux protein, the pump is broken. So if you grow bacteria on a agar plate with the TET, there'll be no colonies. Now, if you add a protease inhibitor that will block the protease activity, now the pump is active and you will get colonies in the presence of tetracycline. So that's a cell-based screen that you can use. Very clever. You can do all kinds of variations of these kinds of screens. So this can all be done at high throughput, tens of thousands of compounds a day. You can screen chemical libraries. You can screen natural products. You can do combinatorial chemistry where you have different chemical linkers that you put together in an array of ways and get thousands of different chemicals. And you can also look at the structure of your target and design molecules that might fit into it, or even use the computer to say, here's the structure of our enzyme. Here's the active site, design molecules that fit into it. We call that in silico screening. And you can do this in high throughput fashion with robots. Here are some robots doing some antiviral screening here. You have these microtiter plates with many wells in them, and the robot fills the wells with the reactants that are needed for the assay. The robot arm then puts the plates into an incubator. After a certain amount of time, they're removed, and the results are sent to the investigator. So, robotic high throughput screening. Now, unfortunately, you get resistance to antiviral drugs. Any drug you have, you get resistance to. Although I must say, I haven't seen any resistance to Paxlovid, which is really remarkable. And that's because viruses replicate very efficiently and they have high mutation frequencies. And so this is a concern in chronic infections where you give people antivirals every day. And in fact, we have resistance to every antiviral drug in the arsenal. We do have resistance to Paxlovid. We can do it in the laboratory, but it's not resistant. SARS-CoV-2 viruses to Paxlovid haven't been found circulating extensively in people. So resistance is a worry because we don't have a lot of antivirals. You get resistance to one, sometimes you have no choice to go to another one. You can't treat with the same drug. If no other drug's available, the patient's doomed. But if you can study resistance, you can get information about the mechanism of resistance and maybe help you design new strategies to prevent it. So the mechanisms are basically mistakes made by the viral polymerases as they copy the nucleic acid. So for RNA viruses, the polymerase, here's an RNA polymerase of poliovirus, which copies the viral RNA. It's error prone. And most RNA viruses have no correction mechanisms with the exception of coronaviruses. That's where there's the Roger Maris asterisk here. Some of you will get that reference. The RNA viruses make one misincorporation in 10,000 to 100,000 nucleotides polymerized, which is about 1,000,000 times greater than our host DNA mutation frequency. So in an RNA genome of 10 kb, this frequency leads to one mutation in 1 to 10 genomes. So if you're making tens of thousands of genomes per cell, you can see that there's a lot of potential for resistance. Now, the nidoviruses, of which coronaviruses are a member. So nidovirus is an order, and there are multiple families within an order. So Coronaviridae and the other Nidovirales all have an RNA polymerase, which is shown here, many different proteins comprising it. The red is the actual catalytic subunit. And there's an exonuclease, which is an error-correcting enzyme. It recognizes mismatches that the polymerase makes, cuts them out so that they can be fixed. But all other RNA viruses, to our knowledge, do not have error correction mechanisms. And so the error frequency is very high. Now, DNA viruses, DNA polymerases do have error correction mechanisms. And so DNA viruses evolve more slowly than RNA viruses because they have less diversity. And the way this works is that the DNA polymerase is copying the strand of DNA. It makes a mistake. It happens. Biology is not perfect. Then this exonuclease cuts out the mismatch and the DNA polymerase can go back and fix it. So DNA viruses evolve slowly, and so you have less drug resistance. Okay, so that's a part one of antivirals. And we are going to next time look into the mechanisms of some examples of antiviral drugs. So you don't want to miss next week. So next week we're going to have the Contagion reveal and antivirals part 2. Oh boy, I like antivirals very much. I think they're very interesting. Let's go back to the questions. Charlotte, hello Charlotte, says, I tried to get Novavax and they said nobody has it. My second shot this year, but nobody has it. Should I get an mRNA? Well, the mRNA is more reactogenic. You'll have more side effects in your arm and so forth. But you know, in your area, you may not have it because there is low availability. It's up to you whether you want to try and find a place that has it or get the mRNA vaccine. People like the Tolstoy quote. Can Mpox be stopped? I think immunization works well. Remember, the vaccine you're using is the smallpox vaccine, so it's not perfect, but it can work. But unfortunately, we don't have antiviral drugs that work well against monkeypox virus. Yeah. The Tolstoy quote sounds much cooler in the original Russian. So maybe if Veronika is still here, you can give that to us in Russian, and not Cyrillic because I can't. I want to read it in the English alphabet. Yeah, smallpox vaccine is effective against Mpox. Comorbidities affect antivirals and vaccine effectiveness. Well, for vaccines, of course, if you have some kind of immunodeficiency, and there are many of them, right? There's not just one. That's a comorbidity that would affect vaccine effectiveness. Yeah, so other comorbidities can affect antiviral efficacy as well. If the antiviral can't get to where it's supposed to go, or if it's metabolized too quickly because you have heightened enzyme activity, then that would affect it as well. I love Tolstoy's cynicism. Feels contemporary, indeed. How about post-exposure puncture tetanus vaccine as another post-event? Yeah, that's good. Of course, I teach viruses, so I don't even think about that. But yes, when I ask the one exception, I'm really talking about viruses. But yeah, that's for sure. That's good. It's a good question. Is combination antivirals effective? Well, for HIV and HCV, yes. We're going to see next time how we use combinations of those to stop resistance, but not for other viruses. Ari Ozone asks, is phage hunting a form of blind screening? I think not, because what you are doing is you're saying, I want a phage that will kill this specific bacteria, which is infecting this patient, right? You do screen various sources for those phages. I mean, first of all, you can go into lab collections and screen those, or you can go into environmental collections, samples. So it is a kind of screening, obviously, but I don't think it's blind because we're specifying the specific bacteria and we know the mechanism, right? The phage is lysing the bacterial cell. So with the blind screening that I talked about, there was no concern for what virus you were using or what the mechanism was. You just wanted inhibition. Visto asks, when an effective antibody is found, how is the sequence to produce that antibody? So when you have cells making, there's all kinds of ways to do this. Let's say you screen patients who have recovered from infection for potent antibodies against the virus, and then you do that. You get B cells from them, and you culture them, and you make antibody. But then you sequence the heavy and light chains of those cells, and because antibodies are made of heavy and light chains, right? Two heavy and two light chains. You get the sequence, and then you make a DNA copy of those. You put them in some cell that will produce the antibody. So basically, you want to produce the antibody in cells in culture, in fermenters, and make lots of it for clinical application. Will, success. I answered rabies. I'm learning. Of course you are learning. You come here every week, man. You're going to learn. You're going to absorb it eventually, right? I'm very proud of you. Yeah, it doesn't matter, but it's a good principle that there are some bacterial diseases that you can limit after infection with a vaccine. Okay. I appreciate that, actually. Yeah. Peter loves the amount of learning that happens during these sessions. You know, I'm glad that you guys learn. It's the whole point, but I learn also, right? I don't remember about tetanus vaccine being given after the bite, or after the not bite, sorry, after the nail stick or whatever. Yeah. Robots. Yeah, those robots putting people out of work. A guy who worked for a pharmaceutical company said he loads up the robots on Friday, and he goes home, and then Saturday the data comes streaming in over the internet, and he can analyze it and design a new experiment for Monday. Isn't that cool? Okay, Visto. Good question. Are there phage that attack another virus, a virus of a virus? In a way. So there are some, they're called virophages because so what they do is they infect a cell that has been infected with another virus, and they inhibit the production of that virus. So so in a sense, they attack it because they reduce the production of the other virus. But what's interesting is that they also need things that the other virus produces. So they take stuff from the virus and then they kill it. Seems to me biting the hand that feeds you, little 9-inch nails, right? But nobody got the asterisk reference. Oh boy. Nevitz wants to know, are the viral mutations purely random, or are there mutation hotspots? Yes, there are mutation hotspots. And what's the driver? Well, like in an RNA genome, there could be a secondary structure that the polymerase bumps into and it slows down, and so it tends to make mistakes right there. And in DNA genomes, there can be similar regions of the genome that make the polymerase pause, or sometimes homopolymeric stretches, like lots of C's or T's or G's or A's, confuse the polymerase, right? Did I put in 4 or 5 or 6, right? So yeah, there are signatures of these hotspots. Can AI help with the in silico screening someday? For sure, because you put all the knowledge. So it's a learning, right? Learning thing. You put all the knowledge about antivirals and their targets, their structures and their targets into the AI. And then you say, okay, here is a enzyme. We don't have an antiviral. Design an antiviral. So we already do that in silico, but AI is going to make it better, right? Because it'll be based on what we know. Yep. Mark wants to know, I've read many of the oncogenic things EBV and CMV do. Any clinical trials? I believe for antivirals? I don't know. That's a good question. Yeah. Let's look. Well, so there are some CMV antivirals, right? Because it's a herpes virus, and so you get like ganciclovir, right? Derivative of, or similar to acyclovir. But there aren't any for EBV. And I'm not sure, I'm sure I have seen research paper people working on it, right? But um, it's kind of a thorny problem because any virus which establishes latency in some cell, the DNA is there. The antiviral will prevent the production of new viruses, but you're never going to get rid of DNA. And that's the problem with hepatitis B virus antivirals, right? So they stop replication, they stop disease, but the DNA is there forever. So you have to take the antivirals forever. And it's the same with HIV, right? So it would be good if you could get one that got rid of the genome. And we talked about that on a recent TWIV. Because the nature of the way the viral genome is suggests some antiviral approaches. John says there was a great discussion on Paxlovid on TWIV, deep dive. I don't know if it was HIV, but it was already existing against, like SARS-CoV-1. And so it was just taken and checked against SARS-CoV-2, and I think it was modified somewhat, right? Angry Penguin writes, UPenn found a right-handed antiparallel double helix inside influenza ribonuclear protein complexes that slides strand against strand as the viral polymerase copies RNA. Can you explain what this means? I have no idea. Let's look it up because I haven't seen it. Influenza, what are our keywords going to be here? Influenza, antiparallel, double helix. Let's try those. Those are relatively unique. So influenza viruses package their RNA into ribonuclear protein complexes that have a right-handed antiparallel double helical structure. The polymerase uses strand sliding to process along the RNA while maintaining this double helical structure. So that's what it means. So the polymerase is sliding along this double helical structure, which is the viral RNA, and copying it. They said it could be a target for future antivirals. So interestingly, we don't have antivirals against we have antivirals against the neuraminidase of influenza. We have antivirals against the endonuclease of the polymerase complex, but we do not have antivirals against the RNA polymerase. And so maybe this could be a target for that. Well, here is very Nika's Russian translation of the Leo Tolstoy quote, and I'm not going to try it because I'll murder it. Vizdrovo. Cool, I like that. Russians usually use shorter versions of this, like, despite doctor's efforts, he survived. I see. So this is the longer version. Oh, so if I put this into, I can't copy it there. Let's see if I can. I want to put it into a translator and see what it translates, but I can't do it. Tona wants to know if a 23-year-old female needs to get vaccinated with HPV if she got 2 shots at age 13 and 14. Okay, so I'm not going to give medical advice, but it depends what vaccine she got. So if she got a vaccine against 16 and 18, she would be set against those two. But there are other high risks. They're less frequent. So you might consider Gardasil 9, which has 9 serotypes in it, right? And get protection against that. It's a good question, Tona. You should send it to Daniel, okay? I want to hear it from his. So this is me thinking logically, but he may say, yeah, you're fine. The other ones are so rare, it doesn't matter. So send it to Daniel, Daniel at microbe.tv. Good question. Yeah, really good questions tonight. Beautiful. Love it. So yes, they're going to start producing Novavax again because they got approval. I don't know how long that's going to take. Are there any antiviral discovery mechanisms that have been proposed but aren't feasible? You mean ways of discovering drugs? I think everything is feasible today. It's just a matter of you being clever to devise the screen, right? I'm not aware of any, though. Um, no, I'm not. Peter S. says automation has its place, and automation of loading lab instruments is a perfect place. So loading the wells, those 1,500 well microtiter plates, you can't do it by hand. It would no human could do that. So the machine does it really well, and then puts it in the incubator and takes it out, records everything. Okay, Ariozone steps up to the plate. When I was a kid, I liked baseball. I don't care a whit about any sport anymore. But when I was a kid in the 60s, you may Babe Ruth had this home run record for the season of I think 56 home runs, which stood for many years. And then in 1960, Roger Maris hit 60 home runs, but the season was 162 games versus 156 of Babe Ruth. Okay, so in the record book, they say home run record per season, Roger Maris, but they put an asterisk because it was in a longer season. So this is the famous asterisk. If you do something and they try and take away your impact, you get an asterisk. So I like to use the asterisk, and nobody gets it, but I do. So I'm happy. Yes, and with RNA viruses, plus stranded RNA viruses, recombination hotspots as well. Okay, what else we have here? Artemis in Switzerland in the 70s and 80s. They were recommending tetanus boosters every 5. Then they decided that 10 was enough. Then I was told I had enough boosters for life. I think I'm still getting one every 10 years. CMV is not oncogenic. EBV is right. Treatment for CMV, none for EBV. Nevitz, so as a student, can I write that down that the polymerase was confused, causing the mutation and get a patent? No, so you can't anthropomorphize. I wouldn't actually take away points for that, even though I tell my students. But in this case, I thought it was a good way of explaining the mechanism, right? So a better way to say it was in a homopolymeric stretch, the polymerase starts to slip and may make misincorporations. So thank you, Nevitz. You're right. Oh, yeah, there you go. Eliza, asterisk for Maris because they said he played more games, hit 61 home runs, breaking Mantle's 60. Right. Oh, the asterisk has been removed. Cool. Yeah, and he was in a race that season with Mantle, and Mantle went to 60, and he got 61 and in 162 games. So, yeah. I referred to rabies as a bacteria. Sorry. I misspoke, obviously, because, of course, rabies is a virus. Yeah. Gary got the asterisk. Guess that dates me. Yep. And here is Angry Penguin's title, A Molecular Basis of Influenza Ribonuclear Protein Complex Assembly and Processive RNA Synthesis. I think I saw that and considered it for TWIV, but I thought it was really too complicated to relate to a general audience. Yeah. Hello, Steve from Kauai. Second time, TWIVO earlier. Got the asterisk too. Do you know of any federal researchers who have lost their jobs? Yeah, I do. People at NIH. Yep. Oh, Babe Ruth had 60? Yeah, that's right. Babe Ruth had 60 in 156 games, and Maris had 61 in 162 games. That's why he got the asterisk. But Ms. Martian was a Sammy Sosa and Hank Aaron fan. Now they're way above 60, right? I don't know how they are, but it's also a steroid-fueled home run. Thank you, Vanity. I'm going to try. I want to see what the translation is. Thank you, Steve, for your contribution to science communication. We appreciate it. We need it to keep these podcasts going. Yeah, that's an inside baseball. And that's the only one I know. Because as a kid, I was 8 years old in 1960. I was in the summer. I'd go to the pool with my friend, grammar school friend, and we'd listen on our transistor radios to the baseball games on the beach. And so we got into this 60, 61 asterisk stuff. Yep. Don't feel bad. Anthropomorphizing is only human. Very clever people we have on these office hours. I love it. I really love it. 165 people, 139 likes. Hit the like button, folks, just to try and attract more people. Welcome, Mark. Good to have you. Legatic gets a tetanus booster every time I show up embarrassed in the ER. Well, you know, if you've had one in the last 10 years and you do something that makes you go to the ER, you don't really need to get another one, right? Funny to hear an educator saying nobody gets it, but I do, so I'm happy. Yes. You know, Rio, when I said it, I didn't actually like that. What I meant was I came up with something that reminds me of my youth, so that it makes me happy that I remember that, not because other people didn't get it. So I as soon as it came out of my mouth, I said, nah, that doesn't sound right. And you picked it up. People are listening. I love it. What makes obesity a comorbidity for SARS-CoV-2? Well, it's a good question. I don't think we actually know, but obesity is an inflammatory disease. And as you know, inflammation is a part of severe COVID. And so somehow those are linked. So if you already have a lot of inflammation and now you have a virus that's increasing inflammation, that there you go for more serious COVID. Did you flip for baseball cards as a kid? Yeah, I did. Yep, I did. Loved baseball cards. I even had a Roger Maris card. I don't have any of them anymore. Here's what I did have. Had lots of pennants of different teams. I don't know where I got them. Maybe 50 different pennants. And I had them in the wall of my parents' house in the basement, right? And, you know, I left. I went to college and graduate school. I wasn't home anymore. And my mother, I guess, got tired of them. She took them all and put them in a garbage can outside at the end of the driveway. She said, you should have seen the people flocking to those pennants. They were gone in 5 minutes. You know, remember the baseball pennants, the things that end in a point and you have on a stick? You go to a game and you buy one. I said, Mom, why did you do that? She said, did you want them? I said, no, not really. Ay. Hoarders. Thank you. You think we're cute. I just want to emphasize that I have not participated in the watching of any sports for thousands of years because I used to love to watch baseball games. And then I said, what am I doing spending 2.5 hours watching other people entertain me? I want to make the content. And so I stopped that and I made my own content, but long before podcasting. Is the usage of, Electron says, great name, is the usage of oncolytic viruses a question of oncology or virology at the end of the day? Well, it's both, right? They're oncolytic viruses, viral oncotherapy. So I guess it's oncology mainly because it's just another anti-tumor drug. You could look at it. You need to know virology to make them, right? But then they're used in oncology. They're going to be. Not only a few have been licensed so far. But yeah. Leo says a decent Burgundy goes with great Twiv. Leo has sent us some very nice wines. Thank you, Leo. Really appreciate it. Yep, Ruth had 60, Roger Maris 61, 1R in Roger Maris. Baseball cards were good for clothespinning to your bike to make motorcycle noise. Absolutely. The crappy cards of the players you never heard of, you clipped and they would hit the spokes of your bike and, oh my gosh, Wayne, I used to do that for sure. I used to do that for sure. Okay, now we're talking baseball. Tom is very proud of the 57 Braves. I was 7. Hank Aaron, Warren Spahn, Eddie Matthews. Very good players. Yep. Rach, I'm awake now, just in time for the end. 2:49 in Isle of Man. Welcome, Rach. We're almost done anyway here. 10 more minutes. I love how every week we have some other tangent that bubbles to the top and resonates with you guys and gals. Like last week it was language, and who knows what it was the week before. Now we're bubbling on baseball and stats and cards. What is the current record? Oh, less meant to ask Google. What is it? Office hours is now This Week in Baseball. Well, Jill, it's funny you should say, but This Week in Baseball was a Mel Allen radio program this weekend. And that's where the name of all these This Weekend podcasts have come from. So I used to listen to Leo Laporte's podcast, This Week in Tech. And he said once that he loved Mel Allen, so he named his podcast after Mel Allen's This Week in Baseball. And I named TWIV after This Week in Tech. So we have a little legacy of respect handed down there. I think that's pretty cool. The single season record is 73, set by Barry Bonds. There you go. Elizabeth writes, my mom threw away all our action figures when we were kids. They would be worth a fortune today. All the original gangsters, Star Trek, Star Wars, Battlestar, Planet of the Apes. Yep. Moms just want to clean up. I kind of collect stuff here, books and stuff that people give me. I should clean up. Yep. Okay, let's see. What do we got here? Okay, everyone responded. We now have 155 likes. Thank you so much. I said that already. Did I have a banana seat bike? No, those were pretty cool. I knew kids who had those. Yeah, those were cool. Truth or dare, I had a shoebox full of cards I flipped with many Mickey Mantle, Whitey Ford, Roger Maris, etc., stolen from my grandparents' attic. I didn't watch any sports. She loves that Twix is named after Mel Allen. Yeah, we're the grandchild of Mel Allen's This Week in Baseball, right? This Week in Baseball, This Week in Tech, This Week in Virology, Neuroscience, Microbiology, Parasitism. I said neuroscience. Evolution. The Twix. We do have a couple that don't do the This Week thing, but most of them are. MN. I put my avocado in a paper bag for 4 days. It never ripened. I read that refrigerating it early will stop the ripening from happening. Hey, MN, that was a topic of like 2 weeks ago. It's okay. Yes, if you had a banana seat, you needed handlebar tassels, right? You needed the big handlebars that went up and tassels coming off of it, yeah. Nevitz Forever had a Casey Stengel baseball card, Mets 65, and thought I hit pay dirt. One is currently listed on eBay, starting bid 79 cents. I'm rich. Yeah, right. Did your mother throw out your comic books? Yeah. I didn't have a lot. She threw them all out. After I left, she threw everything out. And you know what? It was fine because I didn't take it with me, so that meant I really didn't want it, right? Back to virology. Yes. There's controversy in the literature. Many are claiming that the viral spike in CMV hyperactivates PDGF, controversially causing glioblastoma. There is some evidence that there is a side role of CMV in some cancers, right? I'm not up on it, so I don't know. But I do see that. Yeah. Ethylene speeds up ripening. Well, in fact, someone said here, Vanity says, was it with an apple? Didn't say the apple gave off something that ripened the avocado. Yeah, that was Mark Martin who said, hey, you put an apple in with whatever you want it to ripen, and it is ethylene that is speeding up the ripening. It's a gas. Mr. Doreen, hello. I used my brother's baseball cards with a clothesline on my bike to make engine sounds. Oh, the things we did when we were young. That's the key, folks. Do stuff and remember it later. A plant growth factor, Tom tells us. Angry Penguin says, okay, now we're getting back into the ripening thing, the ripening meme. If an avocado is picked too early, it will never ripen, but will only rot. They have to be mature when picked to ripen. Good to know. Good to know. Visto asks, are viruses and their effects responsible for many cancers? 22% of all human cancers are caused by viruses. 22% in numbers. Not just in the different kinds of cancers, right? Just the numbers of people on Earth that have cancer. 22%, like EBV, HIV, hepatitis B virus. There's some polyomaviruses that are associated with cancer. I think there are 11 different human viruses associated with cancer. HTLVs. Okay, Vanity has bought these premature avocados. I never do that. Wow. Tom says, in my first grad school pass, early 70s, I studied plant physiology, plant growth factors, and developmental biology. Well, in the Steinberg home, the fruit are always ripe. You can guarantee on that because of that. That's great. Peter S. says, large supermarkets have cabinets or small rooms filled with ethylene for storing some produce right before it goes on shelf. They're called ripening rooms. I did not know this. And I used to work in a supermarket. I never saw a ripening room. Well, Noir says, I like to remember stuff and do it later. Veronica, the same. And with that, we're going to say Polka. Veronica. I would like to thank the moderators tonight. We've had Vanity Nutrition, Tom Steinberg, Les Fabi. I think that's all. Oh, that's enough. Thank you for moderating tonight. It was a pretty calm night, right, as things go. And thanks, everyone, for coming. Really, really appreciate your presence. When I started, there were 40 people. I said, it's going to be a light night tonight, which is fine. But it turned out to be a normal night. So thank you all for coming. So when are we going to come back? Next week. Next week will be May 28th. Gosh, folks, May is almost over. Before you know it, the summer will be over, and the nice warm weather, which I haven't seen yet, will be over too. Steph was here too, right? Steph was here. I didn't see her recently, but thank you, Steph, also for being here. So next week, we'll have Antivirals Part 2, but we will also have you guys are going to drive some content. You're going to watch Contagion and find 3 scientific things that were wrong. And Vanity, you can do it too, even though you took the course. I don't know. Did I give extra credit? I don't remember. But everybody can do it. And we'll have a lot of fun discussing everyone's answers. Okay? That should be great. Okay, folks, thank you for coming tonight. Until next week, be safe. Bye-bye.